We're moving on now to the second part of our Enterobacteriaceae lecture. And in this lecture, we're going to focus on the organism Escherichia coli. The general features of Escherichia coli, or what's more commonly called E. coli, is that the organism ferments glucose, lactose, trehalose, and xylose. So we already know that this organism ferments glucose because that's one of the key characteristics of all of the Enterobacteriaceae members. But it is important to know some of the other carbohydrates or sugars that the Enterobacteriaceae members ferment. We're going to talk about some diagnostic tests that will allow you to differentiate one from another based on fermentation, especially the glucose and the lactose. So E. coli will ferment both the glucose, because it's an Enterobacteriaceae member, as well as other sugars, including lactose. It is indole positive. The E. coli will ferment glucose using the mixed acid pathway. It is also methyl red positive, Voges Proskauer negative. It does not produce hydrogen sulfide, DNase, urease, or phenylalanine deaminase, or PDA. And it cannot use citrate as a sole carbon source. Now, all of these tests are going to make sense because we will discuss each one of these tests in detail. You know, what it is, why it's done, how it's done, what is positive, what is a negative. You can also look up these tests in your textbook, and the textbook will go into even more detail on each one of these tests. But right now, it's just important to know the major characteristics of each organism, and then we are going to have a separate lecture next week on diagnosis of enterics, and we're going to talk about all of these different tests. Now, the E. coli, there is something called Uropathogenic E. coli. Now, this is a type of E. coli that commonly causes urinary tract infections in humans. So, E. coli is a normal flora in the gastrointestinal tract of humans. So what happens is our normal flora in the intestines will sometimes cause urinary tract infections, especially in females. So in females, you have the urethra that's in very close proximity to the anus, and so you can sometimes get some E. coli that can move from the large intestines. Uropathogenic E. coli has pili. It has an enhanced propensity to attach to the mucous membranes of the urethra and bladder. So it goes into the mucosal cells and the urinary tract, and usually when we urinate, we wash out any bacteria, especially intestinal bacteria that may have been in too close proximity to the bladder region, but the uropathogenic E. coli, due to those pili and that strong ability for it to attach, does not get washed out with the urinary flow very easily. So we also have E. coli, so we have the uropathogenic that causes urinary tract infections, and we also have E. coli strains that are intestinal pathogens. These are the Enteropathogenic E. coli, or EPEC, Enterotoxigenic E. coli, ETEC, Enteroinvasive E. coli, or EIEC, Enterohemorrhagic E. coli, or EHEC, and then the Enteroadherent E. coli. And there are two different forms that are included in the Enteroadherent E. coli that we will discuss. So we're first going to go over the ETEC, or Enterotoxigenic E. coli. These organisms are associated with diarrhea of infants as well as adults in both tropical and subtropical areas. It's much more common cause of infection in developing countries, and it's also nicknamed traveler's diarrhea or turista. It's usually spread through consumption of contaminated food or water, which is why when you travel to an area that you've never been or a foreign country, you're advised to only drink bottled water, don't eat foods that aren't cooked well or foods that wouldn't be cooked like salads since they would be washed with possibly contaminated water. The ETECs have a high infective dose, so you need a larger amount of organism to actually cause disease in an immunocompetent host. So you need between 10 to the 6 and 10 to the 10 organisms. Usually it's a self-limiting infection, and it will generate symptoms such as watery diarrhea, nausea, abdominal cramps, and low-grade fever. And you're going to see that these symptoms are going to be a common theme throughout the Enterobacteriaceae lecture, because these are your typical GI types of symptoms. So the ETEC organisms produce virulence factors, of course. They're pathogens, so they produce two different types of enterotoxins, a heat-labile enterotoxin, or LT, and heat-stable enterotoxin, which is ST. So the LT toxins are the antibodies against the LT are protective, and they act in a similar way as cholera toxin. We haven't talked about cholera yet, but we will. Vibrio cholera is an organism that causes the disease cholera, which is an intestinal disease that causes profuse watery diarrhea. So Vibrio cholera produces cholera toxin. This LT, the heat-labile enterotoxin, is very similar to cholera toxin. There are two fragments, A and B, and A is the enzymatically active portion of the toxin. The heat-stable toxin acts in a slightly different way than the heat-labile toxin. The heat-labile toxin activates cellular adenylate cyclase, which increases ATP. The heat-stable toxin stimulates guanylate cyclase and causes an increase in cGMP. We have another pathogenic E. coli. This is Enteropathogenic E. coli, or EPEC. This belongs to a limited number of O antigenic groups, and the mechanisms of how EPEC causes disease are not completely characterized. There is a plasmid-borne adhesion factor called EAF that enables the organism to adhere to the mucosal cells. And you remember we talked about plasmids in our week 1 lecture, where we mentioned many organisms can take in plasmids, and plasmids are circular self-replicating pieces of DNA that commonly include virulence factors that will give organisms an advantage if they get lucky enough to get one of these plasmids. Now, the clinical manifestations of EPEC, there are outbreaks that are common in nurseries and daycare centers, especially associated with hospitals. EPEC is an infection that is rarely seen in adults. It's much more common in infants. It's also more common in developing countries and includes symptoms such as low-grade fever, malaise or being tired, weak, vomiting and diarrhea, as well as having mucus in the stool. Enteroinvasive E. coli is a dysentery-like illness. It is similar to Shigella. Shigella is an organism we are going to talk about very soon, and Shigella causes dysentery. Enteroinvasive E. coli directly penetrates and invades the intestinal mucosal cells and destroys them. This occurs normally in adults and in children, and symptoms include fever, abdominal cramping, again being weak, tired, watery diarrhea. The E. coli is normally motile, our regular old E. coli that is in all of our intestinal tracts. But the enteroinvasive E. coli may be non-motile and does not ferment lactose. So that is a differentiating feature of the enteroinvasive E. coli compared to our typical normal flora E. coli. We have our enterohemorrhagic E. coli, and the most notorious strain of the enterohemorrhagic E. coli is O157H7. And our EHEC is associated, which is why it's enterohemorrhagic, it is associated with a hemorrhagic colitis, and in severe cases, especially in children, it causes hemolytic uremic syndrome. So hemolytic uremic syndrome, usually individuals that have this will have a low platelet count, they will have a hemolytic anemia, and what happens is they end up having kidney failure, and many times this hemolytic uremic syndrome leads to death. So the O157H7 strain was identified in 1982, and there have been numerous outbreaks associated with O157H7. In the summer of 1993, there was an outbreak associated with eating undercooked hamburgers from a fast food chain called Jack in the Box. In the later 1990s, I believe it was around 96, there was an outbreak in California that was associated with Odwalla unpasteurized juices. So there have been very notorious outbreaks associated with EHEC. The virulence factors of EHEC contain virotoxins 1 and 2. Virotoxin 1 is a phage-encoded cytotoxin that is very similar to Shiga toxin, and Shiga toxin is produced by Shigella. So phage-encoded, if you remember from our week 1 lecture, bacteriophage. The phage are viruses that will infect bacteria. And EHEC also has fimbriae, which will allow the organism to adhere to the intestinal epithelium. The EHEC organisms, as we already said, have been involved in numerous foodborne outbreaks. The symptoms include severe abdominal cramping and watery, bloody diarrhea, which is why it's a dysentery-like illness. There could be low-grade fever or else a complete absence of fever. The stool usually does not contain white blood cells. And how you can identify E. coli O157H7 or your enterohemorrhagic strains, you can use a McConkey agar. So McConkey agar is a selective agar that will select for gram-negative organisms. So in a specimen that's going to have a lot of normal flora, like a stool specimen, you want to try to use selective media to get rid of as many normal flora organisms as possible. So if you take your stool and you grow it on blood agar, you're going to get all of your gram-negative, gram-positives to grow. The only thing you're not going to grow are your fastidious or your fussy organisms. However, if you use McConkey, none of the normal gram-positive organisms are going to be able to grow. Only your gram-negative organisms will grow. McConkey agar also allows you to identify lactose fermenters. Now, there's a special McConkey agar that contains sorbitol as the carbohydrate instead of lactose. And these are sorbitol McConkey agar plates. Now, E. coli 015787 will not ferment sorbitol within a 48-hour incubation period, whereas regular E. coli or non-015787 E. coli will ferment sorbitol. You can also do all sorts of agglutination tests, PCR types of tests, and keep in mind that there are many enterohemorrhagic E. coli strains. 015787 is not the only one, it's just the most notorious one because of all the outbreaks that it's been associated with, but there are many other EHEC strains out there. We have another type of pathogenic E. coli, which is Enteroadherent E. coli. And this is associated with two different types of disease. It causes diarrhea as well as urinary tract infections. And there are two different types of enteroadherent E. coli. There's the diffusely adherent E. coli, or DAEC, and the enteroaggregative E. coli, or EGGEC. So the DAEC is associated with UTI and diarrhea. It's associated with cystitis in children and pyelonephritis in pregnant women, and it's also associated with chronic or recurrent UTI. So individuals that get a urinary tract infection and it seems to go away, and 3 weeks later they get another urinary tract infection and it goes away, and then a month later they get another one. So there are many individuals that get these recurrent UTIs. The enteroaggregative E. coli causes diarrhea by adhering to the surface of the intestinal mucosa. It produces a very watery diarrhea. It can also include vomiting, and as well as dehydration from all of the diarrhea and vomiting. Usually this is going to happen in children, and symptoms can persist for 2 or more weeks. E. coli can also cause extraintestinal infections. So other infections such as meningitis, which is going to be more common in newborns, and also more highly associated with strains that are able to produce a capsule. It can also cause sepsis, which can include bacteremia, can lead to shock as well as death. Can cause pneumonia, nosocomial infections, or hospital infections. And there are also other E. coli Escherichia species. So instead of Escherichia coli, there's Escherichia ermanii, which is a yellow pigmented organism that has been isolated from cerebral spinal fluids, wounds, as well as blood, and can be a source of foodborne infection. And there's also Escherichia vulneris, which is associated with wound infections. And more than 50% of Escherichia vulneris strains produce a yellow pigmented colony. So here you have Escherichia vulneris, which has a kind of a normal pinky morphology on agar, and then you have the Escherichia ermanii that has more of a yellowish pigment, and this is McConkey agar. So McConkey agar is selective for gram-negative organisms, and it will normally show you lactose fermentation. So now we're going to move on to the third part of the Enterobacteriaceae lecture.